right, everyone, good morning. Thank you for that introduction, Rachel. I think I'm going to change my name to Kellan Weston now, forever and ever. Okay, so I'm here to give just a very brief overview. In fact, it's such an overview, I'm afraid it'll barely do justice to either CTNNB1 or to MED13L, but kind of like an introduction to our conference today. Hopefully a little bit of background for those of us who know about CTNNB1 but don't know about MED13L and vice versa. So today I'll just do a quick overview of MED13L, quick overview of CTNNB1, kind of ask the question of why are we having a conference together today and tomorrow? And I'll go over some common phenotypes, and really the reason we're all here together is because of the potential for sort of common approaches to therapeutics. So first off, for those who don't know anything about MED13L, which I confess I really didn't until starting to work with MED13L Foundation. So MED13L syndrome is caused by variants in the MED13L gene. It's on the 12th chromosome, and it's part of this, let me see if I can have a pointer. Perfect. So MED13L, which sits right here, it's this little orange circle-y thing. It sits in as part of this mediator complex. So mediator is this pretty large multi-protein complex that acts as a transcriptional co-activator, and it works together with enhancers to connect them to the different transcription factors, which is all required together for successful transcription by RNA polymerase II to create mRNA transcripts. And so deletion of MED13L or any sort of disruption to MED13L can cause a disruption to transcription of genes, often genes associated with very, very key core growth pathways in your body. And there are multiple different sort of theories for what could be the underlying molecular mechanisms of MED13L syndrome. But kind of a predominant theory, so under normal conditions on the left here is what I just described, where MED13L is working to help RNA polymerase II do its job. But then in MED13L syndrome, there's this sort of disruption to gene transcription. And there's evidence that these variants can be either gain of function or loss of function. So you may actually have disruption going in either direction. And one model is that this causes cyclin C, which comes together with MED13L to be aberrantly released into the cytoplasm, which then leads to mitochondrial dysfunction, decreased ATP production, and increased susceptibility to cell death. Okay, on to beta-catenin. So beta-catenin it has two main jobs. So the first of its main jobs is to sit here at adherens junction. Cellular junctions that are found throughout your entire body. And beta-catenin works together. It binds to cadherins. Specifically, it binds to E-cadherin. So these little green transmembrane proteins here that facilitate this adherens junction between cells. And catenins, specifically beta-catenin, along with alpha-catenin, work together to kind of connect these E-cadherins to the cellular cytoskeleton, here represented in orange. This is the scaffolding of the cell, so it kind of works to really hold your cells together, quite literally. Beta-catenin, even more commonly known and studied, it's a critical component of the Wnt signaling pathway. Don't worry, Wnt stands for wingless. It comes from fly models before this. Clement can tell us all about that later. But beta-catenin, here represented by this kind of pink circle. So under normal conditions, when you do not have Wnt signaling active in your cells, beta-catenin gets phosphorylated, so it gets these little phosphorylation tags by this complex of proteins that includes axin, dishevel, GSK3, APC, and cyclin kinase 1. And this is called the destruction complex. And so once beta-catenin gets phosphorylated by the destruction complex, it serves as a signal for beta-catenin to go and get basically it gets degraded by the proteasome. But when you have the binding of this Wnt ligand, it's a very small ligand to this frizzled transmembrane receptor. This actually recruits the destruction complex up to the frizzled receptor and LRP56, which then preserves beta-catenin, which is then free to translocate to the nucleus, where it interacts with these TCF, LEF family of transcription factors to upregulate Wnt signaling genes. And kind of the product of this is that all of these target genes code for proteins that are, again, key proteins involved in growth and development. MED13L does seem to converge on the Wnt signaling pathway somehow. It's an unknown mechanism. It's yet to be studied. Here are just a few pieces of facts that have yet to be tied together, is that a University of Alabama drug screen done with MED13L Foundation, it gave 2 candidates that do modulate the Wnt pathway, this verapamil, which is a Wnt inhibitor, and plant sterol esters. Wnt was also seen to be upregulated in a MED13L model mouse. And then what Dr. Anna Felser would give in a minute, is that there's some plasma proteomics data from patient plasma that points towards possible downregulated Wnt signaling. So we have some mixed signals here, but there might be some potential for common underlying molecular mechanisms. There are also a lot of common neuronal phenotypes that are seen in models of MED13L and CTNNB1. So one of those common phenotypes can be seen in neurons. So what you're looking at in figure A here is a pyramidal neuron. This is a glutamatergic neuron. This is derived from the hippocampus, a part of the brain which is involved in a lot of spatial awareness as well as many other things. And you can see that this is in a MED13L knockdown mouse. You see a dramatic decrease in this branching, this dendritic arborization. And also you see a decrease in the number of spines, neuronal spines. These are what synapse onto other neurons to form connections within your brain. And you see a similar phenotype happening in this. This is also a hippocampal-derived neurons in a CTNNB1 knockdown mouse. So we seem to have similar molecular dysmorphism happening in the brains of both of these syndromes. There's also as you all well know, especially parents and meeting other parents of kids with CTNNB1 or MED13L, there's a really strong overlap in clinical phenotypes between MED13L and CTNNB1. So I'm just going to highlight a few of them. These are both taken from just the gene reviews articles from each of these syndromes. And we have the most prominent phenotype is intellectual disability and developmental delay. Hypotonia is very common in both of these syndromes, motor delays, vision defects, as well as speech delays. And so these 2 foundations, now that we're working together, we can really work towards common efforts where we're working with 2 different disorders, but we're working together along this same kind of research roadmap path. And in particular, we've got natural history studies. So this is the collection of patient data, especially over time as patients grow and develop, really asking the questions, how does this syndrome evolve with age in this patient population, so that we can start to be able to predict what the outcomes may be for these patients, so that we can also predict what a therapy may be able to target, in a longitudinal manner. We're also doing disease concept models. This is with COMBINEDBrain. This is where we find caregivers, parents, and do an in-depth interview with usually about 10 to 15 parents. And then we transcribe that interview and do an in-depth coding of that interview, picking out core disease domains, such as the domains I just showed you in those 2 graphs 2 slides ago. And this actually helps us sort of cross-validate our natural history studies to really see what matters to parents that may not be picked up in the literature and may not be completely picked up in the natural history studies. Along with that is patient-centered outcome measures. So this is all the different surveys we've been throwing at you all. I'm going to especially plug the ORTIS, the toileting survey, because I'm heading that. And I know that one thing that really often does not get picked up in the literature is toileting abilities, as almost silly as it sounds, but truly, or actually lack thereof of toileting abilities. So we're really trying to develop sort of a survey that really captures what matters to caregivers and families, so that that can actually be used as an outcome measure for clinical trials. And all the different surveys that we have on RareX, Citizens, Simons. If you visit the tables out there, they would love to advertise all of these surveys to you all. And then also biomarkers. We're working together to find biomarkers. So the plasma proteomics study, which Anna is about to talk about, how we actually analyzed our different blood samples all together and sort of did this cross-disorder plasma proteomics. Our transcriptomics study with Unravel, our drug repurposing screen, gut microbiome study, collecting feces, also EEG studies, which are happening upstairs. So all of these efforts, basically everything from IDing the gene all the way out to getting drugs approved, we can kind of work together and collaborate and compare our data. So with that, thank you, and I don't know if we have questions.